Today, we'll be discussing the role of secreted molecules and embryonic proteins in somatic embryogenesis in plants. Somatic embryogenesis is an important biotechnological technique for large-scale propagation of elite genotypes. Identifying stage-specific compounds associated with somatic embryo development can help elucidate the ontogenesis of somatic embryos. Somatic embryogenesis is the process by which somatic cells develop into plants under in vitro conditions. Hormones are known to stimulate embryo formation. Besides hormones, other classes of molecules have also been identified as embryo-stimulating factors. During embryo formation in plants, different types of molecules like oligosaccharides, enzymes, and arabinogalactan proteins are synthesized and secreted into the cell environment. Some important secreted molecules and embryonic proteins that play a vital role in somatic embryogenesis are ribosomal and mitochondrial proteins. These proteins are more abundant in the early stages of somatic embryogenesis than seed maturation proteins. These proteins are more abundant in late stages of somatic embryogenesis. Somatic embryogenesis-related proteins, SCR. These proteins are involved in the initiation of somatic embryogenesis process. Then pentatrichopeptide repeat containing proteins, PPR. These proteins are involved in the synthesis of new proteins related to the growth and development of somatic embryos. Then extracellular proteins. The presence of specific extracellular proteins is correlated with morphology of somatic embryos. Then AGL15. This is a transcription factor that promotes somatic embryogenesis by interacting with other factors, including auxin, ethylene, and racinosteroids. Other secreted molecules and embryonic proteins include auxin, esterified pectins, oligosaccharides. Oligosaccharides means in this cell wall in plants are complex structure of multiple carbohydrates and proteins. In addition to its role as structural framework, cell walls serve as an extracellular matrix that contains signaling information for plant growth and development. So oligosaccharides derived from the plant cell wall and have been identified in identified in conditioned medium in the different cell suspension cultures, including spinach, maize, tobacco, rice, etc. It is generally thought that the production of oligosaccharides in culture medium is the result of carbohydrates or glycoprotein splitting by cell wall enzymes. The resulting oligosaccharides might contribute to the nutrition of the developing structures, but also to the control of the embryogenic process. Then peptides. Signaling peptides have been identified recently in plants, like phytosulfokines, PSK, are the peptides that are disulfated 4 amino acid peptides. First, this PSK isolated from the asparagus cell suspension culture medium. PSK enhances cell growth and substantially increase the number of PSK binding sites, indicated that PSK and this receptor like kinase act as ligand receptor pair. Then arabinogalactan proteins, AGPs. The AGPs are a family of proteoglycan with very high carbohydrate content, 90 to 98%, and consisting of high levels of arabinosyl and galactosyl residues and branched structures. In addition, their protein core is rich in hydroxyproline, serine, alanine, glycine amino acids. Then pathogenesis-related proteins, PR. The enzymes, PR proteins that have been detected in the cell culture media are mainly gluconeases, chitinases, etc. In embryonic suspension cells of barley, 3 chitinases and 1 beta-1,3 glucanase have been isolated. So it has been hypothesized that the culture medium accumulates this PR proteins may be responsible for degradation of the callose in the cell wall of embryonic cells. Other molecules like vesicillins, Rs20 proteins, proteins of the RAB family. Then role of secreted molecules and embryonic proteins. I will discuss the role of secreted molecules and embryonic proteins with the help of the results of a recent study by Bertini et al. 2021. Bertini et al. Studied the stage-specific protein regulation during somatic embryo development of Carica papaya golden. According to the results of this study, a total of 801 proteins have been reported in the somatic embryo at globular, heart, torpedo, and cotyledonary developmental stages. The globular stage presented a higher number of unique proteins, like 16, and 7 were the isoforms of 60S ribosomal proteins, suggesting that high translational activity at the beginning of the somatic embryogenesis. Then proteins related to mitochondrial metabolism accumulated to the high degree at the early developmental stages and then decreased with increasing development. Means they contributed to the cell homeostasis in the early somatic embryos. During embryo maturation stage, a progressive increase in the accumulation of vesicillin, late embryogenesis abundant proteins, and chloroplastic proteins were observed. Protein metabolism is very intense in embryogenic cells. Therefore, protein metabolism represents a key event in the early development of somatic embryos. During the early developmental stages, means globular and heart stage, several proteins of RAB family were detected, like RABA5A, RABA5B, and RABA6A, being unique in somatic embryos at the globular stage, and RABE1C being unique in the heart stage. Two members of RAB family, RAB2BV and RABH1B, were also detected only in the globular and heart stage and were not detected in subsequent stages like torpedo and cotyledonary stage. Then two enzymes, GST-F13 and catalase 1, were also unique in somatic embryos at the globular stage, as these both function in antioxidative pathways in response to stress and have been repeatedly been identified in proteomic studies of somatic embryogenesis. These findings suggest that antioxidant enzyme, means GST and CAT, play important role in stress response during the development of somatic embryo. With respect to the somatic embryos at the heart stage, RBT4B protein was unique. Then for somatic embryos at the torpedo stage, only ARF1 was identified as unique protein. This protein is necessary for recycling of pin-formed auxin transporter, PIN, that mediate the directional transport of auxin between cells and for the functioning of auxin-mediated developmental pathways. Arabidopsis ARF1 mutant showed an impaired auxin response gradient that leads to embryo patterning defects and arrested growth. Then, consistent with the importance of auxin carriers during development, ARF1 is also responsible for establishment of auxin gradients that determine the polarity of procell embryo and embryo axis. Thus, the accumulation of ARF1 in the torpedo stage suggests that this enzyme plays a crucial role in auxin transport, which ultimately leads to the differentiation of somatic embryo. At the final developmental stages, XYLA protein was unique to the cotyledonary stage somatic embryos. XYLA catalyzes the isomerization between D-xylose and D-xylulose, which are polysaccharide components of the cell wall of somatic embryo cells. This enzyme has been previously detected in this Caricapaea somatic embryos in the cotyledonary stage and during the final step of maturation, and also in sugarcane embryonic calli. So accumulation of XYLA in somatic embryos exclusively at the cotyledonary stage suggests that this enzyme may be play an important role in mediating carbohydrate and polysaccharide sources for developmental process such as cell wall formation and cell growth at the end of the somatic embryogenesis process. Thank you.